Hello everybody, I am Frédéric Baret from INRAE in France. I will present a study called High-throughput estimates of the radiation interception and use efficiency in wheat crops. This study has been conducted with Shoyoung Liu, Marie Weiss, and Benoît de Solan, and Pierre Martre from mostly INRAE as well as PPRC, so the Plant Phenotyping Research Center in Nanjing. Wheat is one of the main crops cultivated. It is the third crop cultivated in the world. It is grown under a very wide range of climate, soil, and management conditions. Improvement of yield is mainly achieved through two main processes: the total amount of biomass produced and the harvest index. The harvest index is the yield divided by the total biomass produced. So currently emphasis was put on harvest index by breeders, but limit seems to be reached soon. Between 50 or 60% of the total biomass can be transferred to the grains to make the yield. So the only way to improve the yield is to improve the total amount of biomass produced by the crop. And so breeders probably should pay now more attention to increase the total amount of biomass produced. Plants are mostly capturing light to make biomass from the photosynthesis processes. So it's capture carbon dioxide from the atmosphere and water from the soil, and then form the sugars to release oxygens. Since 1977, a very simple efficiency model can describe the accumulation of biomass. So the accumulation of biomass is mostly the total amount of photosynthetic active radiation incoming and intercepted or absorbed by the canopy, multiplied by the light use or radiation use efficiency. So in this simple equation, you have at least two efficiency factors. One is the radiation use efficiency, RUE, and the other is radiation interception of absorption efficiency. In this case, the fraction of absorbed photosynthetically active radiation. So the radiation use efficiency from this simple equation can be measured by computing the ratio between the total amount of radiation absorbed by the canopy divided by the total amount of biomass. The fraction of absorbed radiation depends on illumination conditions and canopy architecture. So the illumination conditions are mostly described by the derived fraction. So it's 0 when there are clouds in the sky, and it's almost close to 1, or let's say 10% or 90% when it's mostly coming from the sun under clear sky conditions. And of course, the position of sun also is very important to account for. Regarding the canopy architecture, there are two main variables or parameters that need to be accounted for. One is the average inclination angle, and the other is the green area index. So the green area index is defined as the amount of the area of green elements per unit ground area. It's expressed in square meter per square meter of elements, green elements per square meter of ground. As you can see on these figures, the fraction of absorbed radiation depends for a given sun direction on the green area index and on the average inclination angle. It increases with the green area index, reaching a maximum value close to 1 for green area index of let's say around 3-4 green square meter of elements per ground square meter, and it also increases with the leaf inclination for relatively high zenith angle of the sun, and it decreases with the average inclination angle for relatively low sun angle. That means sun angles that are quite close to nadir directions. Of course, there is a large variability of illumination conditions between days, but between locations as well, and also during the day. You have here for wheat at the tearing stage an example of the evolution of the fraction of absorbed radiation with its different components. So the direct components, the one coming from the sun, the contribution from the sun, and the diffuse component, which is here, of course, stable. The fraction is stable across the day, but the fraction of direct radiation increases here on clear sky condition with time in the morning, and then decreases in the afternoon, as compared to a cloudy condition where the direct component is almost zero, and the diffuse component is contributes more to the total amount of radiation. You have here another example, the stem elongation stage. So later in the growth season, where the fraction of absorbed radiation is almost saturated, so close to 1, all the radiation is absorbed by the canopy. And of course, in this case, the importance of the canopy, of the illumination conditions is not very important. It is very important to get accurate FAPAR values to better understand the biomass accumulation. We have here some results from model simulations. It's the serious quality crop growth model that simulates the accumulation of biomass or the functioning of the canopy depending on the climate and the soil and the management techniques. The simulation have been done here for 5 locations, from high latitude sites in Finland at 61-degree latitude down to low latitude locations in Sweden at 14-degree latitudes. And you can see here, depending on the way we describe the light interception or absorption in the model, you have here 5 different ways to compute the light absorbed or intercepted by the canopy. And you can see here in relative values of biomass accumulation for the whole growth season. You can see very large differences, almost 40% or 45% in some specific conditions and specific models. So this is very important conditions. How can we measure FAPAR, the fraction of absorbed radiation? So one usual way is to make a radiation balance. So we measure the incoming radiation, we measure the reflected radiation, we measure the transmitted radiation, and we measure the radiation that is absorbed by the soil, by the background, which is the transmitted radiation multiplied by the fraction that is absorbed, which is 1 minus the fraction that is reflected or by the canopy. So the fraction of absorbed radiation can be computed using this simple equation. And you can see here the variation of the different terms. So the transmittance is dropping with the increase of Grignard index. The reflectance is changing, is decreasing slightly, but not very much with the growth of the wheat. And as a result of this equation, the FAPAR is increasing a lot with the growth, the development of the leaf area, and then with some saturation here close to 0.9, and then a drop with the senescence. You can see here that the drop is not very important. It's not very important because all the elements are contributing to the fraction of absorbed radiation when you measure the balance, the radiative balance. But actually, when we want to compute the fraction of absorbed for photosynthesis, it's important to make a difference between the green and non-green elements. And in these techniques, it's not possible to make a distinction with green and non-green elements. Further, this technique is low throughput because you need to put small sensors in each of your microplots. You need to repeat those sensors to have many replicates of those sensors because these are small sensors and they are not very representative. And as I told you previously, there is the impact of the senescent leaves at the bottom of the canopy that is not accounted for. So for this reason, we prefer to use another proxy of the fraction of absorbed, which is the fraction of intercepted radiation. You can see here on those simulations that the fraction of intercepted radiation is a very close assumption, is a very close approximation of the fraction of absorbed radiation. For low chlorophyll, even for low chlorophyll content values, when the leaves are more reflecting and transmitting more light, so probably contributing more to multiple scattering, the difference here is very small between the fraction of intercepted radiation and the fraction of absorbed radiation. And this is due to the fact mostly that the leaves are strongly absorbing radiation because of the strong absorption of the chlorophyll and pigments. And these techniques, when measuring the fraction of intercepted radiation from the top of the canopy, so looking downwards, allows to avoid accounting for the senescent leaves. So taking just the green leaves into account, and the green leaves, as I said previously, contribute to the photosynthesis mostly. How can we model the fraction of intercepted par radiation? So as you can see, the architecture depends of the canopy depends strongly on the way the leaves are oriented in the space, and mostly how they are inclined. And you can see here that when we measure precisely the distribution of the inclination of the different elements, these distributions follow what we call an ellipsoidal distribution. So it's a distribution of the facets over an ellipsoid of revolution. So it allows to, with a single parameter, which is the average inclination angle, to describe the whole distribution of the elements. The other aspect that is very important is how the elements are distributed in space. So if we compare the actual distribution of the leaf of wheat and the stems of wheat in space to a turbine medium distribution, where the elements are randomly distributed in the canopy volume, in terms of the fraction of intercepted radiation, it makes almost no difference. You can see here the difference between the relationship between the fraction of intercepted radiation and the green area index in the case of the turbine medium, which is a solid line, and in the case of the 3D model, which represents a very realistic case of wheat canopy architecture. As a consequence of that, we can compute easily the fraction of intercepted radiation as 1 minus exponents of g of the function g, which is called the projection function, as a function of theta, the sun position, the average inclination angle described by an ellipsoidal distribution function, and the green line index, and you divide that by the cosinus of the sun inclination zenith angle to account for the path length in the canopy. GAI and the average inclination angle can be derived from green fraction. As a matter of fact, the green fraction in a given direction is the same as the fraction of intercepted radiation in the same direction. So we can use the same model to relate the green fraction or the fraction of intercepted radiation to the average inclination angle and the green area index. This is illustrated over a given example here for wheat by the relationship between the green fraction at a given 57 degree, where it's no more dependent on the average inclination angle, and the green area index. And the green fraction can be derived from several sensors. It can be derived from high-resolution RGB imagery by classifying the green pixels. It can be derived from multispectral imagery using, for example, vegetation indices that are calibrated to estimate the green fraction using those empirical transfer functions. We can use as well the LiDAR 3D point cloud by eliminating the background using the height measured by the LiDAR and eliminating the senescent elements using the intensity of the return laser beam. What is the optimal observation configuration to derive GAI, the average inclination angle, and as a consequence, the fraction of intercepted radiation from the green fraction? So in this case, we consider that we can estimate very well these with just two cameras oriented in two different directions. So we made radiative transfer model simulations with a Turing medium assumption to identify the best couple of directions to derive green area index, average inclination angle, and the corresponding fraction of intercepted radiation. For green area index, we see that the optimal configuration is a camera looking at 30-degree inclination and another one looking at 60-degree inclination. For the average inclination angle, we need a camera inclined at 15 degrees angle and the other at 60 degrees. But when we are combining the two to estimate the fraction of intercepted radiation, we see that it's much better to get a camera at 0 degree inclination and the other close to 45 degree inclination. So the optimal combination appears to be 0 and 45 degree to estimate the FIPAR. And this is also optimal because at 0 degree you can estimate other traits. So of course you can have the cover fraction. You can have also you can estimate the chlorophyll content and other traits. And at 45 degrees, it's quite interesting because you can estimate, for example, probably you can count the plants at early stages, and also when you have relatively small microplots, small width, then if you have very inclined directions, it's quite difficult to make the measurements because the observations will be contaminated by the other microplots close by. These findings were validated from 3D model simulations using the Adelwitt architecture model that allows to derive a 3D very realistic description of the canopy architecture and its dynamics. That's why it's 4D. It can be coupled with a Raytron, which is a rendering machine that allows to derive, to compute the images corresponding to these scenes, same images as a RGB camera could get on the same scenes. And you can see that to estimate the green area index, it's quite efficient using the two directions, 0 and 45 degrees. It's a bit less accurate for the average inclination angle because because the green fraction is less sensitive to the average inclination angle. But when we recombine the two green area index and average inclination angle into the fraction of intercepted radiation, we get a very nice and very accurate relationship between the estimated one and the reference one. So we can get very accurate estimates of FIPAR. This has been also validated over actual experiments. This is for the green area index, when you can see a very close relationship between the reference measured in the field by destructive measurements and the estimated from the green fraction at 0 degree and 45 degrees. And finally, for wheat crops with radiative balance measurements in the PAR during the first stages where there is not too much senescence, you can see also a quite fair relationship between the estimated values from the two directions and the one estimated from the measured values by the radiative balance and the estimated one from the measurement of the green fraction in the two directions. This means that we have a very good consistency with the previous theoretical results. The green fraction can be measured with several sensors and systems. It can be measured with handheld systems like LiDAR, with sensors on a tractor. It can be measured with Phenomobile, which are fully automatic robots, or on gantry systems. You have here examples of the green fraction evolution during the growth period for wheat, during different years for two modalities, one irrigated, one non-irrigated. And you can see that we can derive quite nice dynamics of those green fraction. This green fraction can be transformed into estimate of green area index and average inclination angle. You can see here at the same for the same years and same dates the evolution of the green area index and the average inclination angle, and these two variables can be combined to compute the fraction of intercepted radiation at the time of the measurements with the phenotyping systems using the same simple equation here. And as a consequence, we can compute the heritability of the green area index, the average inclination angle, and the fraction of intercepted radiation over different experiments. So this has been done in Gréoux in France with an experiment with 40 to 20 cultivars, 3 replicates, and 2 modalities. And you can see that we have very high values of heritability, generally higher than 75, 0.75, and in some cases very close to 1 actually. So it's quite heritable traits. So if we recap, how can we estimate the fraction of intercepted radiation and then estimate the radiation use efficiency? So the radiation use efficiency can be, as I said earlier, estimated by computing the ratio between the total amount of radiation that is intercepted by the canopy during a period divided by the biomass accumulated during this period. So we start by the measurements here, for example, with the Phenomobile of the green fraction at 0 degree and 45 degree. From that, we estimate at the time of the measurement the green area index and the average inclination angle. We interpolate with time those measurements to get continuous estimates of the green area index and average inclination angle. And then we combine them together in this equation to get a fraction of intercepted radiation, and this continuously during time. That means every hour in the day and every day in the period. And for that we need, of course, the estimates of the direct fraction and the position of the sun. That depends on the latitude, the day in the year, and the time in the day. Once we have got the FIPAR, we can combine with the amount of incoming PAR every hour, and then accumulate integrate that over the period to get the total amount of intercepted photosynthetically active radiation, and then by dividing by the total biomass, compute the radiation use efficiency. So finally, with this technique, we can measure the radiation use efficiency. These are some examples here of the radiation use efficiency expressed in gram of biomass per megajoule of intercepted radiation. So we can see for those 6 cultivars that we have difference between cultivars as well as differences between modalities. So the problem of estimation of the radiation use efficiency is that we need high throughput biomass measurements or estimates. So biomass currently is mostly measured with the classical method by destructive sampling, which is quite interesting because it has no bias, but because it's very expensive, the spatial representativeness is not very important because we have small sampling area. It's low throughput, and of course it's destructive and invasive, so it's difficult to repeat that during the season many times on microplot, otherwise there will be no more material in the microplots. So we have direct measurements maybe with robots, but the problem is that it's not yet existing. Such systems are not yet existing. It could be medium throughput and probably quite expensive. But the advantage is that there is no bias. And what has been developed is indirect methods based mostly on allometry, where we measure or estimate the surface from high throughput measurements, from that estimate volume, and then make estimate of the mass by making some assumption mostly on the density of the elements. So this would be high throughput, non-destructive, probably affordable, but it needs always to be calibrated. So we need to make at least a few measurements of biomass to calibrate the empirical relationships. And probably we could have possible bias because the density may not be the same depending on genotypes or conditions. So as a conclusion, we propose a scheme to compute green area index and average inclination from green fraction. The green fraction could be easily derived from several systems, as we demonstrated. When we combine green area index and average inclination angle, that can be accurately derived from the green fraction at 0 degree and 45 degrees. We can also estimate the fraction of intercepted radiation using a turbid medium assumption that applies well to wheat crops, and from that estimate the radiation use efficiency if the biomass measurements are available. Unfortunately, we need high throughput and unbiased estimates of biomass to provide statistics on the radiation use efficiency, to investigate the variability of the radiation use efficiency, and to compare the radiation use efficiency that is modeled by the crop growth models, to compare that with actual observations in the field, and probably improve the crop growth models by taking into account more interactions between stress modules, or taking into account the germ type, probably germ type variability in the radiation use efficiency. So I thank you very much for your attention.